Welcome to Broad Eye, the podcast that explores knowledge gaps in ophthalmology and eye care. Hello, everyone, and welcome to another episode of the Broad Eye podcast. My name is Sean Maloney, and today I will be speaking with Dr. Yves Sauvé. Dr. Sauvé is a recent retiree. He was an associate professor in the Department of Ophthalmology and Visual Sciences at the University of Alberta. Dr. Sauvé, welcome to the podcast.
Well, thank you, Sean. It's very good talking with you.
Thank you. We've spoken in the past, actually in another podcast about, I think it was about 5 years ago. So I'm happy to have you back. And
happy to be here. Thank you.
Good. Well, I enjoyed that conversation then, but I'm going to try and grill you with a lot of different questions this time. And a lot of these are coming from personal interest, things that I don't understand, and things I also hear people bring up a lot. So I think we can have a lot of fun with this. But I was hoping we can, kick it off just talking a little bit about your journey. Now that you're recently retired, maybe just talk a little bit about your academic journey and maybe touch on how you got into the realm of vision and specifically vision and aging.
Sure. I started with a bachelor in biochemistry at Montreal University, and there was one class about neurochemistry that really interested me, and I said, I want to do a master there. So I did a master in neuroscience, and then after that, I got an interest in growing nerves, and I heard a talk by Albert Aguayo, who was a very, very important neuroscientist in the 80s and 90s in Canada. He funded the Network of Excellence in Neuroscience. So I approached him, and he said that the actual slide that got me interested wasn't from him, but was from Michael Raspinski. He says, go see him, but he doesn't take any PhD student, just so you know. So I went to see Mike Raspinski, and I said, I'm very interested in how you're able to take a sciatic nerve and replace the optic nerve with that. And not only that, but you put an electrode in the brain of the hamster, and you're able to record visual response. The optic nerve has been completely transected, and the suture on the stump there, anastomosed, is that sciatic nerve. And I say, I want to do that. And he says, well, I don't take any PhD student. I say, I know, Dr. Aguayo said so, but can I attend? So I attended the research and the experiment. And Sue Kierstad was a postdoc. And basically, she taught me everything and let me do the experiments myself. And at some point, I took a sheet of paper and I gave it to Dr. Raspinski. And I said, this is where you sign. And he says, where? He says, it's just there at the bottom. He said, okay. And he signed, and I was his PhD student. So to make a long story short, there was the visual system, Sean. There was the retina that was stimulated with light, and there were responses in there. And then I was looking for a postdoc. After I finished with that, there was this guy in London who was doing really interesting therapy in retinitis pigmentosa, which you know obviously about RP. And he basically was putting cells in their RPE cells in the back of the eye, and he was able to preserve. So that was a preventative approach. Anyway, he needed an electrophysiologist, someone to record response from the brain. I said, well, I'm the guy. I can do that. So I did 6 years of beautiful years in London, UK, where I was recording responses. And there was a postdoc there, Jean Lawrence. She said, I'm going to teach you about the retina. And when you're done with your postdoc, you're going to know the retina inside out. So this is how I got interested in the retina, Sean. Before, I was just abusing of it to prove that we could grow nerves and to replace the optic nerve. But now I was really, really fascinated by that multi-layered structure. So that's what I've done, transplants. And it took me to Utah, Salt Lake City after that, where the lab moved from London. And I was doing something called ERG, electroretinogram, recording responses from the retina itself. So I was really sucked in by that beautiful organ called the retina. And it was just getting better and better. Dr. Ian McDonald from the University of Alberta, 2005, recruited me to continue working with the electroretinogram and different therapies. And then we moved to Stargardt. And this is where I started studying Stargardt, macular degeneration, and therapy with preventative with DHA, which is an omega-3. So this is my story.
It's interesting, and Dr. McDonald is a common or a mutual contact, and he's been on the podcast recently, and he also highly recommended having you as a guest. So there's certainly a history there.
I have the highest respect for Dr. McDonald. The honor that he did me by headhunting me in the depth of Utah was just wonderful.
I don't know if you know this, but so I met him back in 2008 or 2009 when I went to the NEI when he was there to participate in the iGene project to get my mutation sequenced. So or determine the mutation in my condition. So that's when I met him a number of years ago. So
so you were there for almost 2 years. Yep.
Yeah. So you mentioned DHA, and maybe you come back to that. But I was wondering if you can talk a little bit about the retina, of course, and just retina with age. So, what changes as we age in terms of our, in terms of the retina in general, and how would you classify? Like, there's certain changes that would just be normal associated with aging, I would assume, and not necessarily pathologic, and there's others that would be pathologic. So maybe if you can just comment on those a little bit, please.
Yeah, with pleasure, Sean. I invite you to a little journey here. People, if they want, the people listening to this podcast, if they feel comfortable, can close their eyes, because we ultimately see with our brain. So we can close our eyes and have mental pictures. So if you feel comfortable, close your eyes, and we're going to imagine that light, we are light, and we are entering the eye. So we enter the eye by the cornea. And the cornea is the first structure that the light hits. And with age, it gets a bit drier. So you'll see people using Sustain or different lubricant because of dry eye. And DHA can actually help with that. And we are light, we continue our journey, and then we arrive at the lens. And at the lens, with age, two things happen. The lens will become a little bit less flexible. So we can't see near things very well without wearing reading glasses. That happens at the age of about 45 years old. That's the lens. And the lens with older ages will become opaque, slightly opaque. So that's cataracts. But it does become opaque anyway, just slightly. And we don't really notice if we don't have cataracts, but it does become more opaque. So that's age. And of course, this is the major reason people go blind is cataracts. But we can reverse this. We can replace the lens with the intraocular lens. So that's good news. These are all good news when it comes to the front of the eye and age and diseases. Most of them are treatable. Now we keep our journey. We got the jello-like structure called the vitreous. And what happens with age with the vitreous is we get more floaters with age. So that is little objects that move in your visual field. I don't know if you are able to see floaters, Sean, with your condition, but floaters are increasing with age. And then we move to the retina. And the first layer we see is ganglion cell and their axon. Axons is the arm of a neuron. So a ganglion cell is a neuron, and it is the only output to the brain, the only output. And unfortunately, with age, there's a little bit less of them. They decrease in number. And then we have bipolar cell that allow the talking between photoreceptors. We're not there yet, but between photoreceptors and ganglion cells, they allow that transmission. So there's a need for bipolar cells to communicate between photoreceptors and ganglion cells. So bipolar cells also decrease with age. especially the one carrying the rod signals. So now we keep moving. We're really going to the back of the eye, and we reach the photoreceptors, and that is the most affected by age. The rods for night vision and for non-color information die in the mid-periphery. So this might be a bit of the reason why people will tend to bump into things in the dark, because that's for movement detection. We'll get back to movement detection later. So the rods die and the cones are preserved, except if you have a disease. But we're not talking about disease. We're talking about normal, healthy aging. And behind the photoreceptors, we're really reaching the end of the retina here. It's not part of it. It nourishes this, is the RPE, retinal pigment epithelial cells. So retinal pigmented epithelial cells, they are monolayers, a single layer of cells, and they are essential to nourish the photoreceptors. Unfortunately, they tend to die, they tend to become dysfunctional, they tend to not nourish the photoreceptors as much. So they are the culprit of why photoreceptors die as well. So that's pretty much with age. I'm gonna talk to you, Sean, about the catch-22 situation, if that's okay.
Of course, yeah. Go ahead.
Okay, so here's our catch-22 situation. Is in order to see, we need DHA, and we talked about it. We talked about that omega-3. It's highly fluid. It's Mother's Nature invention for a antifreeze. So salmon, mackerel, cold fish water, cold water fish. Sorry about that. Cold water fish have that as a natural antifreeze. So they are in water sometimes that goes below zero. And so they don't freeze, they get DHA. It's very fluid. So in order for us to see, we use 50% of all the fatty acid as DHA in the photoreceptors to make reactions of transforming light into electrical signals in a very fast manner. And it's all membrane-bound events. So cells have membrane, and the membrane of photoreceptors is uniquely fluid. So 50% of all the fat in there is DHA. The problem is, so we need that to see. The problem is that DHA is the most oxidable fatty acid in the living kingdom. So it is the most oxidizable. So basically, you need it, but it's going to get oxidized. So you need antioxidant. And as we get older, the oxidative damage, these are free radicals, increase. And there's oxidative damage everywhere in the retina. So exposition to light causes that as well, because light can also lead to damage in the outer retina. So that is our situation. This is Mother Nature favorized vision very much, but the level of oxidation is very high. And the reason is, to make matters worse, is that photoreceptors are not vascularized. They don't have a single blood vessel. It's like when you take your cell phone, right? You don't want to have blood vessels in them, because the camera is going to get a distorted picture with the imprint of the whole vascularization, if you get my drift.
Yeah, for sure. Yeah, makes sense.
You get all these blood vessels in the way, so you don't want that. So how do they get their oxygen? They get it through the RPE, which nourish them, and from a capillary bed called the choroid. So this is the highest level of oxygen you will ever encounter in the human body, is in the outer retina. So these very, very vulnerable photoreceptors, because they have DHA and it's extremely oxidizable, are exposed, this is catch-22, to the highest level of oxygen in the body, because they rely on passive diffusion of oxygen. So in order to make it go there, you need to pump it up very high. So this creates opportunities for diseases, because it's a vulnerable. So age-related retinal disease is often linked with that oxidization of the retina, of the photoreceptors. So we got antioxidants. Well, that's great. So you heard of antioxidants, Sean, and the one that come up are lutein and zeaxanthin. So you can take antioxidants. The problem is, for antioxidants to be there, they need to bind to receptors. And very unfortunately, with age, the number of receptors decreases. It's a natural event that the carotenoids, so these are the antioxidants, receptors, binding proteins, go down. And that was shown by Paul Bernstein from the Moran Eye Center in Utah, showed that. So yes, you can supplement, but there is a maximum amount, a limited amount that can bind because the receptors are going down. And when you look at the picture of a retina, and I'm sure you've seen a retina before in photos, Sean, it's kind of darker in the middle and it gets browner. And that's because of the presence of these antioxidants creating that color. But it gets less brown, less orangey with age. So that is more or less what I had to say about the aging retina.
Oh, there's so many points I want to go with this. I'll respond to a couple of things you had mentioned. Wondering if I see floaters in my condition. My vision is far enough gone now that I don't really see floaters anymore, but I did used to see floaters, so I can certainly relate to that. I like how you brought up DHA, and it just makes me think of so many things. I've read some studies where there was a correlation shown between the amount of these macular pigments and progression of Alzheimer's disease, or dementia. And I'm just wondering whether or not these carotenoids play key roles in dementias. I know there's been some data out there. I actually have seen some data that's currently under review in this space, which is pretty interesting. But the other part of that, talking about having DHA and how critical that is. I'm wondering if, or maybe and maybe don't. I don't know if there's any link in the brain as well, and that's maybe why, or one of the reasons why you need DHA and brain health. I don't know if there's anything in that as well.
I am, Sean, not aware of the link between the antioxidants and Alzheimer itself. But what I do know is that the changes you see in the brain take place in the retina. So these beta amyloid and these senile plates that accumulate in neurons also happen in the ganglion cells. You will see these beta amyloid increasing. But whether this is modulated by DHA intake, I don't have an answer for that. What I know is that there's recent discoveries of a way of detecting Alzheimer, because this is an accessible part of the brain, the retina is, and you can image these plaques, senile plaques, these beta amyloid increase. You can image them in the retina as a biomarker of Alzheimer.
Yeah, that's wild when you think about it, right?
Yeah.
Now, just going on DHA again, and this is only my own curiosity, but when we talk about these carotenoids, so for lutein, zeaxanthin, and then more recently we talk a little bit about mesozeaxanthin as well. The affinity, I guess, for these or the receptors that will bind these in the macula, that is influenced by or can be improved, I guess, that affinity by supplemented with DHA. Is that correct?
No.
No. Okay.
No, I didn't say that, and I don't think DHA does improve the affinity of the CBP. So they're called carotenoid binding proteins. So carotenoids because of carrots. So carrots is full of beta carotene, which basically is an antioxidant and a precursor to other antioxidants.
Okay, no, so okay, so thanks for the clarification on that.
Yeah.
So I guess this leads to another question. I'm going to jump around a little bit. I had queued up a few questions before we were talking. I sent you some questions. I'm going to jump around a little bit, but um, the we're talking about fish oil and these macular carotenoids that we can take supplements.
Yep.
What are some of the maybe in addition to these, what are some of the modifiable factors that are really within our control to when it comes to vision loss or preventing, slowing that aging process
Sure.
that would naturally occur?
Absolutely. So what we can not change, obviously, that's an oxymoron, is aging. So that's the major cause of a lot of conditions like AMD, glaucomas, diabetic retinopathy. But what we and our genetics. So there are genetic predisposition for certain diseases, and in your case, it's a monogenic condition that you have, and it's one gene that causes it. In case of AMD, it's many mutation called polymorphism. So they're not per se disease causing on their own, but they're predisposition genes. So the genetics and aging, definitely we can't change. But what we can change is stop smoking. So smoking increases oxidation in the back of the eye. So we talked about that being deleterious and increasing with age. So you don't want to add insult to injury by smoking. Exposure to light. So when it's a sunny day, you want to wear sunglasses because that also causes damage at the back of the eye. Nutrition. You want to avoid processed food because it contains a very, very low level of the ratio of the omega 3 to 6. So there's way more omega 6 than omega 3. So the culprits there are soy, where you got 7 omega 6 for 1 omega 3, and corn. So corn, you will find 47 omega 6 for 1 omega 3. So I challenge you to find processed food that does not have corn derived products. So purifying elements from the corn itself is really where the problem is. Eating corn on the cob is fine, but as you have a high fructose corn syrup, that really is not a good idea. And corn oil, that's not a good idea. So nutrition. Now you might be surprised by this one, but this one is well established and it might be hard to guess, but education. It has been found that with age, like AMD, you find less AMD in educated people. So we don't know what it is, but education, very, very, very important factor in visual losses that can be modified by the environment. So I can't tell you more, Sean. Your guess is as good as mine, but education is important and it's been proven. And finally, using visual aids, as you might be doing, also is a modifiable factor. You can use them like magnifiers, etc. But yeah, these are the modifiable factors that I can think of at the moment, Sean.
No, and that makes a lot of sense. The education one is certainly interesting, and the first thing that comes to mind, which is probably not correct, is people who have access to higher education or get higher education maybe end up ultimately with higher incomes and can afford higher quality food and not always buying processed food. That might be one element of it, but I could be way off in left field on.
No,
I'll leave that analysis to the researchers.
That's a good guess.
It's just that's all it is for me. It's just a guess. I haven't read about that.
Yeah. I concur, Sean. I concur with what you said.
Okay. So we're talking about changes in the retina, and some of these changes, like any other changes in your body with age, are we can try to slow them down. Some of them are that's just the way it's going to be. Are there any changes in the brain? And honestly, I have no clue. This is just a question that I thought about. But are there any changes that happen in the brain with age? I guess whether those are normal or pathologic that might affect vision.
Absolutely. Absolutely. So talking about the cortex, which is how we generate the reality that we experience, the visual reality is modulated by the retina, but is generated. So this is why I said close your eyes. Oh, I forgot to say you can open your eyes again after our journey. But the journey that we took together through the eye was a generation of our brain. So the brain areas that are responsible for creating visual scene, there's over 40 of them. And 40 distinct areas in the brain that are involved in generating the visual scene, the visual experience. And they're not all affected equally. What has been found is that the one involved in movement, motion, detecting movement and motion are affected first. So this might explain why with age, people tend sometimes to bump into things just naturally, just not grasping the whole scene, the peripheral vision. So movement is detected in the periphery, Sean.
Can I jump in with a question on that? Just because it's something that comes to mind, and I'm probably going to forget it because I'm getting old. So when you're also talking about normal aging and some of the changes in the retina, we talk about loss of rod cells in that mid-peripheral area. So do you think that
it's connected?
Yeah, it's connected. Like maybe the loss of the rod cells are leading to decreased activity in the area of the brain for detecting, motion and spatial awareness.
I was asking myself that same very question, but there are controlled experiments done in monkeys in Oregon, where they showed that if you record responses from these cells in an area called MT of the brain that is responsible for vision, or also called V5. You can electrically stimulate that region through the retina, and you bypass the problem that might be occurring with rods. You will find that the responses are actually more sluggish, not finely tuned to specific orientation. So the detection, so what the brain likes is bars of different orientation. When you record from the brain, if you want a stimulus, you use bars of different orientation. And when you do that, it's just less tuned. So that can be the defect in the rods. That has to be the tuning properties of the cortical neurons themselves, which is generating detection of movement. That's a cortical process, Sean, the tuning. So that is the brain.
Well, that makes sense. Okay, no, that's I just was having just a question.
That's absolutely fine.
Okay, so I thought maybe we could jump into something a little bit oblique, I guess. It is talking about teaching and mentoring. You seem to, even on the call before we started recording this call, you and I were chatting, and the first thing you had mentioned about, you recently retired, but the thing you said you missed the most is teaching. So maybe you can just comment on that a little bit, and why were you so passionate about teaching? Not that it's bad. I'm just curious that not everybody is as passionate about the teaching component of their jobs if they're a researcher.
I want to get started. Thank you for that great softball.
I try to serve softballs if I can, right? The hard ball's coming next though, so just be ready for it.
I'm always ready. So I was thinking when I do research that we don't always find something. We spend a lot of time discovering things perhaps that don't seem to be so useful. And I say, what's the point sometime in doing all that we do, and we find little things that we don't find use for it? And I said, it's a culture. It's a culture of inquisitiveness, and of respect of others, and collaboration. And I think this is preserving that culture. And that can be done also. It's a natural extension to do it by teaching. So when you're in class, you really, really promote inquisitiveness and curiosity and venturing. No venture, no gain. So if a student says, how about this, how about that, I encourage the student to really challenge me, to challenge concepts, to ask questions, as you very astutely did when you ask about orientation. Could it be rod-based as opposed to just the cortex? That's what I like, is to be challenged and together as a group to find answers. So that motivates me greatly. And then I like to make jokes. So I have a bit of a stand-up kind of comic sidekick here. And that's what I like in class, is to engage students via humor, via different concepts, via everyday example. And yeah, I come alive. I come alive. When I'm done with my class, I feel somehow rested, somehow at peace. Yeah, it really, really energizes me so much, Sean.
So how are you going to replace that in retirement? There's a question. There's the hardball for you.
Well, the good thing is there's always opportunities to still teach classes as a sessional. So you can go and teach the class that you want without anything else. So you can focus on that class. So that's what I will be doing, hopefully next year or in the second year, but sessional. So that's my plan, is to give classes still. Yeah, yeah. So it's not over.
Fair. And then you can also do things like this and sharing your knowledge with a global vision care community via podcast.
Yeah, I
probably
appreciate.
from
I
the comfort of
appreciate
home,
very
probably,
much.
right? There you
Very,
go.
very timely opportunity. Yeah, very timely. Yeah, that makes my day, Sean.
Great, good. So I want to just maybe touch on one more thing, which is completely different than anything we've talked about. This may be a softball, maybe a hardball. I'm not really sure, but it's talking about your book. You have published a book recently called "Feeling the Meaning of Life." So I just wanted to know a little bit about that's the genesis of that book. Where'd the idea come from? What was it like writing that? How long did this take, etc.? I'm very curious.
Yeah, thank you for, yes, absolutely, a softball. It's coming from a class that I put together in 2009 with Dr. Karpinski, who unfortunately is no longer with us. But he said, Yves, let's do a class on sensory physiology. And because vision is one of the most important senses in his view, and in my view, no pun intended, is 50% of the class, so 12 lectures based on vision itself. And as I was giving that, the thought came how really, really the emulation of reality is done by the brain, that the brain really generates what we experience. So we can experience distorted vision by fooling with the brain using psychobilin, for example, which is magic mushroom, which is hallucinogenic, or using LSD. That has been proven in an experimental setting to actually change activity of the cortical cells that generate images. So, and people with schizophrenia can see things that are different. So really the brain ultimately decides what you see. And that started everything for me. And I said, how did we get there? How did we arrive to the point that the retina only modulates? Of course, it's super important because it modulates so we can survive and reproduce. So the evolutionary pressure that led to how we see really interested me. So the book talks about, first chapter is how we actually see. So it describes the process. And then it goes to evolution. How did we get there? And in a nutshell, I'm going to try to summarize, is that as we evolved from single cells, there was input and output. So input is the senses. So there were very, very primitive eyes and primitive just light detection in a single cell. So a single cell can detect light depending on the type, and it can react by moving its cilia in the same direction of the source or away from it, depending on whether it's a threat or whether it's actually a goodie that's going to promote my survival. So it's all about survival and reproduction. But we're not talking about reproduction yet because these single cells just can divide themselves. So it's not sexual reproduction. But as we move to more cells, so it took billions of years, so much more years from one cell to many cells than the explosion of life, which took 700 million years. So 700 million years to make all the multicellular animals on Earth, but it took 2 point something billion years to move from one cell to many. So the survival and reproduction to be ensured was very complicated when you move from one to many, because now what you do is you specialize. So one cell specializes in receiving the stimulus, and another cell is specialized in the motor output, okay? Input, output. Now you're gonna make things more complicated. You're gonna interpose cells between, and you're gonna develop nervous systems, like in the squid, which is very primitive and also very, very advanced in terms of nervous system. Anyway, so you get interneurons. So what they do is they make a motor program. They plan the action. And as we evolve, humans and other animals with cortexes, is that there was no longer a need for the stimulus and the output. The interneurons that create that program became free of input and the need for output, which led to rumination, which led to anxiety and depression, which led to the creation of external agents like belief in God, belief in external agents, if you see where I'm going with that. And what I want to say is that these interneurons, we don't feel them. We don't feel their activity. We can feel, of course, when we stimulate our senses, we can feel that, our skin, our five senses, but we don't feel the interneurons firing. And that is a threat. Where's that coming from? It's not comfortable. And there's that region of the cortex called the amygdala, which receives stimuli and decides whether this is a threat or not. And if decides that it's a threat, it goes to the cortex. But it also receives information, the prefrontal cortex receives information about threat that has nothing to do with stimuli, with sensory stimuli. So I'm trying to see the foundation here of why we can ask, what is the meaning of life? We ask that as a fringe effect, a side effect of developing these premotor program. I know it's very abstract what I'm saying, but I'm trying to have a positive spin so that we don't worry. In conclusion, in the book, what I talk about is how do you deal with these false threats, these rumination, this hamster running in the wheel, and creating scenarios and ruminating on the past, is by grounding yourself with sensory stimuli, such as mindfulness. So breathing and feeling the air through your nose, that grounds yourself. That sends the information to the brain, and whatever the thoughts are telling you and revving, just become what they are, a pure creation of the brain that has nothing to do with the sensory reality we live in. So that's more or less my book, Sean. I'm sure you have questions, so fire away.
There's questions, comments. So a couple things come to mind when you're describing that. One is, I remember reading somewhere about one of the explanations, at least, for why so many people have a fear of public speaking is that, the only time throughout history where somebody would be in an open space with a lot of eyes looking at them would have been, somebody maybe who was hunting gathering, and there could have been a pack of wolves or something looking at them in the dark or whatever it may be. All these, dozens or hundreds of eyes looking at them, and the normal response at that time was fight or flight. So that might be one of the reasons why people have, and there's probably, hundreds of these types of examples, right? I guess that's kind of a vision one.
No, but yes, you're right. When we think of the first humans, 100,000 years ago, or 300 to be precise, in Chad, they found Homo sapiens there. But what happens then after that is not so much evolution, not so much mutations, not many changes, very, very minor. So you're absolutely right to think of the first humans, because that would have decided the evolutionary pressure, the mutations that allowed survival and reproduction. We have to consider what their environment was then, and how we have so much modified our environment that it doesn't match with how we've evolved. Genetically speaking, we're misadapted. We're never evolved to drive a car. We never evolved to give public speech with hundreds of eyes looking at us. We haven't evolved to do that. So there might be some misadaptation.
It's funny. I actually really enjoy public speaking, and I also wonder if that's because I can't see my audience. Right? There might be some truth to that. I mean, I know like I gave a speech a couple of years ago as a fundraiser, and there's like 500 or 600 people there, and it was this black tie event, and I really enjoyed it. I found it really, really pleasurable and really fun to be on stage and having that conversation. But I couldn't see anybody in the crowd, so maybe that has something to do with it. But
I think what has to do with it, you're a passionate man, Sean, and you want to share your passion with others. This is why you do these podcasts. It's because you want to learn and you want to share your passion. So that's your driving force. And
I think
that trumps the nervousness.
Yeah, I don't know. I see. This is secretly just this is secretly really selfish, right? I just want to have conversations with people who are a lot smarter than I am, and they don't want to have those conversations with me unless it's like, oh, hey, well, there's going to be thousands of other people listening to it, then it's okay. But
we have a different perspective. I'd say we just have a different perspective.
There you go.
Not smart.
One of the things you had said was about what I liked, and I've seen this before, is the idea of grounding yourself with sensory experiences. So I have a son who had some anxiety and attention deficit when he was younger. Still does, but had a lot more anxiety when he was younger. And one of the recommended ways to deal with that was, it was actually through my sister, who's a cognitive psychologist, who recommended this, but it came from somewhere. I think it came from practice. Was think of focus on 3 things you can smell, 3 things you can see, 3 things you can hear, 3 things you can taste, 3 things you can touch, something like that, to really ground yourself, right? I think that's a common practice in psychology. But
you stabilize, you basically let the thoughts be, and you're no longer a slave of the thoughts. If that makes sense.
It makes perfect sense. I mean, it's sometimes it's easier in theory than in practice, especially we are emotional human beings, but it's good to have that in the toolkit. But
I can divulge, I'm personally anxious as a person, and I have a hard time sometimes just trying to just do mindfulness. It's not coming as easily. So just doing sports sometimes helps, because when you do sports, you really, really depend on your senses greatly. So that can help as well.
That's fair. That's fair. One more question for you. Where can we find your book?
Oh, that wasn't planned. That wasn't planned. You gave me
Why?
six
No.
questions.
Well, I threw a few more than 6 at you, but this is not. No, I'm interested. I think people are listening to this. People are listening to this. I mean, they have to let them know where they can find it. You can't leave them hanging in the wind, and they're going to get bombarded with emails instead.
Yeah. So you can find the book at Amazon. Basically, if you look "Feeling the Meaning of Life," it's available on Amazon.
Perfect. I will. We'll put some links in the. We always publish a blog article with these podcast episodes. We'll put some links to that for everybody. So,
for anybody who would be a French speaker and listening to this podcast as a French speaker, I translated the book in French called "Ressentir le sens de la vie," which is also available on Amazon.
Excellent. There you go. So you had to outdo everybody. You have to put it out in two languages right away.
Two languages.
That's great. Well, there'd be one for me and one for my wife. So is it a sorry?
It is sciencey. But as I write at the beginning and the preface, is that you're allowed to jump sections, and so you can go straight to chapter 5, which is really feeling the meaning of life. And you can go to other chapters that talk about ignorance and convenience as the two major threats in our society: ignorance and convenience.
That's interesting. Well, we'll have to check it out, and we'll definitely link to that in our show notes. So, Dr. Sauvé, this has been fun. I always enjoy speaking with you. Hopefully at some point in the future we can meet up and do this again and do it in person over a coffee.
I would love that.
Yeah.
Give me a shout if you have an event in person. I could try to make my way. Where are you based, John?
I'm just outside of Montreal. So you're familiar with Montreal.
There you go. So, I was raised in Montreal, so it'd be a pleasure to meet you in the flesh. Yeah.
Excellent. All right, Dr. Sauvé, thanks so much for joining me today. It's truly been a pleasure.
Most welcome. I'm very honored to be on your podcast. Take care.
Thank you. And that concludes today's episode of the Broad Eye Podcast. If you enjoyed this episode, please be sure to subscribe on your favorite podcast platform. Of course, ratings and reviews are always welcome. And you can certainly share this episode with any of your colleagues or friends who might enjoy it. Thanks for listening.